Kia ora and good evening. First of all, thank you so much to the Neurological Foundation for hosting us this evening and supporting our work, and thank you to you for coming and listening. We are really passionate about our work and we love any opportunity to talk about what we do. So I hope you learn something and that you find it interesting. So just to give you a little bit of a background about what we're going to talk about tonight, I'm going to give you a bit of an introduction about why we study maternal mood and behaviour. So my group, we're all focused entirely on understanding the changes that take place in mood and behaviour in parents. I'm going to talk about why do we care about this, and then probably a harder question is how do we study this? And I'll talk a little bit about how we approach this question. Then I'm going to talk about some of the recent work that we've been doing that's given us new insights into how hormones are acting in the brain to drive changes in a mother's behaviour. I'm then going to hand over to Jamie and he's going to talk about the Neurological Foundation project that he has just started. And then he's going to end on a question where we thought we should really address, well, what about dads? So most of the talk today is going to be about mums, but we don't want to forget dads, and there's actually some really interesting changes that are taking place in their brains as well when they become parents. So when we think about being pregnant, we often think about the physical changes that take place in a mother's body. So obviously the growing baby inside her and the physical changes that accompany that. But some of the most dramatic changes that a woman will talk about are changes that take place in the way that she feels and in the way that she behaves. Some of these are really positive changes in emotions. She'll talk about this incredible love that she has for this being that she hasn't even met yet. Delight in that child. She might talk about this real change in priorities. She no longer cares that her house is messy where she once would have. Feeling incredibly protective of these children. She'll feel this nesting urge. She wants to go and renovate the house. I did that with one of my babies. And also about how incredibly rewarding she finds spending time with her baby. But alongside that, it's important to acknowledge that this transition into motherhood is not easy for many women. Someone will also talk about feeling at times sad, detached, anxious, frustrated, overwhelmed, worried, and I think every single parent feeling really tired. And in fact, it's been found that in New Zealand, around 14% of new mothers will struggle with a maternal mood disorder, either during pregnancy or in the time that follows after pregnancy. And we're interested in, well, how is the brain driving these changes in emotions and behaviour? And I also want to acknowledge that maternal mood disorders don't just affect a mother, they affect an entire family. So they affect other children, partners, grandparents, support networks. And in New Zealand, this equates to around 8,000 New Zealand families that are impacted every year by mental health issues in new mums. So why do we have this problem? Well, what's contributing to really making progress in this is, first of all, we don't understand how the brain is changing to bring about these differences in mood and behaviour in mums. Another really big barrier is the stigmatisation around mental health, especially in mums, the pressure to make life look all perfect when in fact it might not be. And the last issue is that we have very limited treatment options, and this again comes back to the fact that we don't understand how the brain is driving, this has got a delay, how the brain is driving these changes in the first place. So the goal of my research is to understand how mood and behaviour is regulated in parents, so we want to understand the basic mechanisms in the brain. And by doing that, we want to assist in developing some novel effective treatments and also by sharing our research help to destigmatise the area of parental mental health. So some really beautiful work has come out in the last couple of years around the world that's actually looked at big broad-scale changes that take place in a mother's brain when she's pregnant. So they've done some MRI studies that have tracked a woman's brain throughout pregnancy and into the first couple of years after she gives birth. And what's interesting is there's actually these big changes even in the volume of the brain when a woman is pregnant. The volume actually shrinks right down when she's pregnant. But what goes up, this is called white matter. These are like the connections between different brain regions. And suddenly these connections go way up in the brain of a pregnant woman. And what we think is going on is there's a lot of fine-tuning that goes on in a mother's brain to equip her to be able to deal with the demands of parenthood. This is one of my favourite images that's been taken in people here. This is an fMRI, so it's showing changes in the activity of brain regions in an individual. And whenever it flashes and lights up, that's where a brain region becomes really active. And they've done some work to show that when mothers look at pictures of their babies, the bits of the brain that drive reward behaviour get really active, specifically in mothers. This doesn't happen when they're not mothers, but when they are mothers, if they even look at a picture of a baby, it also happens if they hear their own baby crying, suddenly these regions in the brain start working. But there's actually very little research that's been done to understand human maternal caregiving behaviour. And the reason for this is obvious, that it's actually very hard to do experiments on parents. You can't have a look inside the brain of a woman and find out what's going on. So how then can we make progress about understanding what drives mood and behaviour changes in mothers? And where we can make the most progress is by looking at examples in the animal kingdom. Now, parenting occurs in a large variety of vertebrates and invertebrates. It occurs in insects and fish, amphibians, reptiles, birds, and mammals. And every single mammal on this planet is involved in maternal care. They all look after their offspring. And I'm not going to talk about it so much tonight, but what's interesting is it's the same hormones and it's the same pathways in the brain that are driving these changes in behaviour in all mammals. So it's really highly conserved. And also what will be relevant for what Jamie touches on a little bit later is 10% of mammals are biparental. So that means in 1 in 10 mammalian species, the dad also is involved in parental care as well as the mum. And obviously humans are an example of that, but actually as we'll talk about later, so are mice. So I'll put up some of my favourite examples of motherhood from the animal kingdom. This one here, this is a deep sea octopus that was observed brooding her clutch of eggs for 4.5 years. One clutch of eggs, what they did, this was off the coast of California. They had some researchers that were monitoring this deep sea trench for 25 years. Every 3 months they would go down in their submarines and have a look. And they observed this octopus and she stayed put with one clutch of eggs for 4.5 years. So she would clean them, she would protect them, she would keep them oxygenated. Why did she do that? Well, they discovered that when those eggs are hatched, those baby octopi were far more developed than ones that have a shorter brooding time. So by investing in that time, she had equipped those offspring to be more likely to survive once they hatched. This one here, this is matrophagy. This is a bit gory, and I'm glad we don't have this in mammals. This one here is where the mother actually gives herself to the offspring, literally. The offspring eat the mother. She sacrifices herself so that her offspring can become stronger and healthier. And compared to these related species that don't have this form of matrophagy, these offspring are far more likely to survive. Cheetahs are a really interesting one. So cheetahs, they have really high mortality rates of their cubs. So around 70% of litters don't survive. In cheetahs, the reason for that is predation. So their major predator is lions. So what cheetahs do is when they're pregnant, they go off and they live in isolation. She'll give birth, and then every single 4 days, she moves her whole lair to a new place, and she carries her cubs with her. The reason to do that is that it removes the trail of scent from that mother so that the lions can't get them. And then the last one, this is a more local example. I really like this example of cows. There's this article, it was in the Rural News Group from New Zealand, talking about safety around cows, and everybody knows that you've got to be wary of cows when they've got their calves, because normally they're fairly docile, but when they've got calves, they can be quite aggressive because they're highly protective of their young. And what we can see in every one of these examples is females acting quite oddly, because before they were pregnant, before they'd have young, they never would have acted like this. But suddenly, when they become mothers, their behaviour has radically changed. And this is a fundamental point. Becoming a mother means that behaviour must change. But unlike humans, animals can't do a pregnancy test or an ultrasound and tell them that they're pregnant. And these behavioural changes often take place when she's pregnant. So how does an animal know that it's pregnant and it should behave differently? And the key thing that's communicating to the brain in that animal that you're pregnant is its hormones. So hormones, these are these chemical messengers, they travel in the blood and they direct the function of a whole lot of organs in the body, and crucially they direct the function of the brain. Where are these hormones coming from? Well, there's a gland below the brain, the pituitary gland, that's really important. Ovaries and testes also produce these hormones. And one that's most important and people often don't realise, in pregnancy, the placenta. We think of the placenta as being important for nutrient exchange, getting rid of waste. But one of its most important functions, especially in our eyes, is actually it pumps out huge amounts of hormones. And those hormones travel in the mother's blood and they change the way that she thinks and the way that she behaves. So what do these hormone changes look like? This is just showing, this is in women, this is across pregnancy, when she gives birth and then when she's lactating. These are some of the key changes in hormones that drive behaviour. And the key point is that many hormones change during pregnancy and then in the postpartum period. And it's not that they just change once, they constantly change. So there's these very distinct patterns. You're even getting changes in some of them on like an hourly basis. So we're not seeing constant levels of hormones, we're getting them fluctuating all over the place. And the ones that I've spent my career looking at are hormones. One is prolactin and the other is placental estrogen. These are actually the same hormone, they're just produced by two different tissues. One's made by the placenta, the other's made by the pituitary gland. And this hormone was identified many, many years ago, and it was called prolactin because it's prolactation. It's the hormone that drives milk production, so you need it in order to be able to breastfeed. But the work that we've been doing is alongside it changing milk production, it's also acting in the brain to drive behaviour as well. So how do we study the role of hormones or prolactin in maternal mood and behaviour? I just want to acknowledge my very awesome team. So this is a very talented group of researchers that all look at different aspects of parental behaviour. And we asked two questions in my group. The first one we ask is, well, how does the brain regulate parental mood and behaviour? What we want to know is which bits of the brain, which cells, how do they connect together, how do they work together to change behaviour? And the next question is, well, how do hormones then act on these brain regions to change this behaviour when it needs to, such as when a woman gives birth? And we look at this in two different ways. One is we just look at what is happening in a healthy pregnancy. So we just want to understand basic biology changes. And then we also use models of maternal mood disorders to try and understand, well, what is happening when we're not getting these healthy changes in mood and behaviour. So we use mice to look at maternal behaviour, and as I said earlier, you might think, what's mice got to do with humans? But actually it's got the same hormones and the same pathways in the brain that are driving these changes. So if we're going to use mice, we need to know what does their maternal mood and behaviour look like. Well, they nurse their young, they carry them, they groom them, and they show great motivation to care for their young where they wouldn't normally. They also have a range of changes which we don't think of directly caring with the young, but are actually also really important. One of that is metabolic changes. So A female needs to eat more when she is pregnant so that she can keep up with the demands of breastfeeding. That's hugely energy costly, and so she's got to change the way that her body works to be able to make this milk. They'll also show reduced anxiety when they have young, and this means that if a pup wanders off, that female will go out and get it. Or if the food sources are scarce, then she'll venture a bit further than she would normally to get that food. They'll also show this really protective behaviour, which is common in all mammals, and they'll also do this nesting behaviour. So I've got an example here to show you what this looks like. So this is a female. She has had her pups. Over on this side of the cage here, you can see she's got 3 little pups. This is a barrier that we've put in this cage. It's a very climbable barrier, it's pretty low. And what we see is a female will climb over there, she'll have a look at the pups, she'll lick them, then she'll pick them up in her mouth, she'll carry them back to her nest, and then she'll go back and keep doing it. Now I find this really fascinating because if you take a female when she's not a mother, when she's not in this state, she'll climb over that barrier, but she'll never pick up a pup and take it back to her nest. She will only do this when she becomes a mum. So how do we look at how the brain is controlling these behaviours? If we think about the brain on a whole, it's incredibly complex and it just makes it too hard to be able to look at if we looked at the brain in a whole big system. I kind of liken it to having an entire village made out of Lego and you're being told to replicate it and you don't even know where to start. It's too big to be able to understand. So what we do is we break it down. First of all, we have a look at neurons, the individual cells in the brain, and try and understand what are they doing in a female. And I kind of think of these as individual Lego bricks. Then we take these individual neurons and we try and see how do they connect to each other within the brain. We call these neural circuits. How does one neuron connect to another? And I kind of think of this as like taking your Lego bricks and making a small component of your entire village. Say you might want to make a fire engine. And then from these neural circuits, we can start to build up how these neural circuits together make a brain. And what we're trying to do is write the instruction manual. We're trying to figure out, well, what is the instructions that's forming these circuits so that a mother's brain will function the way that it does. And the way we approach this is we look at, well, what are the hormone-sensitive neural circuits that drive a mother's mood and behaviour? So we're looking particularly at the bits of the brain that can sense these hormones that are changing, the ones that come from the placenta. How are these ones important for mood and behaviour? And I'm going to just tell you a little bit about some of the work that we do in this. So first of all, we break it down to that individual building block. We want to know what are individual cells doing, neurons doing in the brain. And we spend a lot of time at looking at one region of the brain. This is the MPOA, or the medial preoptic area of the hypothalamus, which is a mouthful. But this is the control hub of parental behaviour in males and females in every species. This is a super important little region of the brain for driving maternal behaviour. And it's full of these neurons that can sense the hormones that are coming from the placenta. And so what we can do is we can look at, well, how does the activity of these neurons change? In this video here, these are taking individual cells that can sense this hormone, and every time they flash, the brighter they get, the more active those neurons are. And here we can look at them under a microscope in a slice of brain tissue. This is like a living slice of brain tissue from a mouse. And we can see how these neurons are active in a mother compared to an animal that's not a mother. But fundamentally, this is what's happening under a microscope, and that's not what's happening in real life. A female was walking around and interacting with her offspring. So we want to be able to do this, but in a whole animal. We want to understand how are these neurons, these little building blocks, changing when a female is interacting with her young. And so to do this, we actually use some really advanced technology. We used fibre optics to be able to record the activity of these cells. It's a technique called in vivo fibre photometry, and we can measure the activity of living cells while an animal moves around. And I'm going to show you what this looks like. This female here, she's actually attached to a fibre optic here. This is a really lightweight, flexible cable. We can attach it, detach it. The female doesn't care if it's on. She will happily eat, she'll sleep, she'll give birth with that cable on. But what it means is we can now look at the activity of these cells. So, I'm going to show you here. This is 2 times the speed if you're thinking, why are these pups kicking like crazy? Some pups are put in that cage, and over here, we've got the activity of these cells. When it's down the bottom, they're really quiet. They're not doing anything. But whenever she goes to interact with one of these pups, we get these huge surges in activity in the brain. These neurons that can sense this hormone are getting super active whenever this female interacts with her pups. When she goes away, it drops down. When she goes to get another pup, it goes way back up again. And what we have found is that these hormone-sensitive neurons are actually only active when a female interacts with her pup. You can throw other things at her. You can give her a Cocoa Pop. Mice actually love chocolate. You can give her a Cocoa Pop. They're not active. When they are active is when it's a pup, and especially when it's her own offspring. So that's the neuron. And we've kind of said, all right, we're taking these individual building blocks, and what we're doing is we're establishing that instruction manual of how they're connecting to different regions in the brain. So we're now doing experiments where we're taking specific neurons that go from one region to another and trying to understand how these circuits are controlling behaviour. Here, for example, in purple, we can actually label the beautiful neurons that has sensed these hormones from one region and they project to another. We can map them right out in the brain and figure where they are. We can also record their activity while a female's wandering around. And again, this is looking at the activity of these neurons. Here, they're really quiet. You give them like a toy, which they normally find really exciting. They're not interested. But if you give these females pups, these neurons and these circuits are getting super active. And so we've used this technique to try and understand which bits of the brain get really excited when a female is showing normal changes in mood and behaviour when she's pregnant and when she gives birth. And then what we've gone on, and I'm not going to give you the specifics for this, is instead of just watching these neurons, now we make them really active. So again, using fibre optics, we can activate these cells in a virgin female. So a female that's never had pups, she's never seen pups before, we can now take these neurons or these neural circuits, we can make them have the same kind of excitedness as a mother would. And what we see then is actually we can make a female that's not a mother behave just like a mum, just by harnessing those neurons in the brain that sense those hormones. But one of our key questions, well, how important though is the hormones themselves within these brain regions for these changes in mood and behaviour? And this is that control hub of the brain, the medial preoptic area. There's a few little black dots. This is just a female. She doesn't have, she's not pregnant, she's not a mother. And a few of these cells, these neurons can respond to this hormone. This here is what it looks like when a female gives birth. Suddenly, huge amounts of these cells respond to these hormones. She's got high levels of these hormones pumping through her blood. They're getting in the brain and they're really acting on these neurons. And some of the work we've done is we've stopped just these ones here, these cells, these neurons in the brain, from being able to sense prolactin. So they can't pick it up. It's still going through the body, but they can no longer tell that it's there. If you do that, then mother mice entirely lose interest in their pups. They'll completely abandon them when they give birth. So these hormonal changes are absolutely critical in driving a mother to want to look after her young. So that's an example of where we can stop hormones and the mother doesn't show these normal changes in mood and behaviour. Some of the work that we've been doing more recently is looking at models where we're not getting these healthy changes in maternal mood. So one of them is a dietary stress. We use a high-fat diet. The reason for this is that obesity in pregnancies associated with many complications, and one of those is a greater risk for postpartum mood disorders. And what we can see in mice that are obese is they also have disrupted maternal care towards their pups. And when you look more closely, they actually have disrupted hormone production. Their placentas don't form properly, and they don't produce the same amount of these hormones that are activating the brain to tell the mother to look after her offspring. So we're really interested in unpacking that and understanding, well, why is this happening, and what could we do to get this increase this activity in the brain? And the other one that Jamie will touch on in a moment is environmental stress. So the model we use is we reduce the resources that a mouse has available to build a nest. And I really like this model because I think it's really applicable across many mammals, but also relatable to people as well. And if you just reduce the resources she has to build that nest, then those females show disrupted care towards their pups. And there's actually long-term effects on those pups. They will grow up to have impaired responses to stress for the rest of their lives as well. So we're interested in looking at that model and saying, what are hormones doing, and how is the brain not responding the way that it should in these instances, and what can we do to help? And I'm going to hand over now to Jamie to talk about his current project.
Kia ora koutou. Good evening, and thanks for coming along. Around 2.5 years ago I joined Rosie's lab, and during that time I've mostly been working on males, and I'll touch on that later on in the talk. But after a little while realised the potential we had, and to look at the impact of postnatal anxiety on parenting behaviour, and the tools that we had that might help us to target a potential therapeutic avenue. So starting with the project, thanks to the Neurological Foundation for their funding, this came about as a collaboration between Rosie, myself, and a colleague who focuses on the neural regulation of stress. Just to recap, so 1 in 5 women in New Zealand will suffer from anxiety during or following their pregnancy, and there's a lack of treatment options available. So to address this, we need to better understand how the brain is regulating anxiety in mothers so we can hopefully target treatments. Just to recap, Rosie's talked about the medial preoptic area in the brain, sensitive to prolactin. And I like to think of it as the central hub that orchestrates all of these behaviours that we've been talking about. The medial preoptic area, prolactin-sensitive neurons project to many areas in the brain. And one we're particularly interested in for this project is the PVN. The PVN contains corticotropin-releasing hormone neurons, which are an integral part of the stress response. This is where June Kim comes in. June studies the neural circuitry controlling stress in mice. And June found that in the PVN, there is a projection from the MPOA that regulates the HPA axis, the hypothalamic pituitary adrenal axis. Now the PVN CRH neurons release CRH, which stimulates the production of ACTH from the pituitary, and that in turn releases cortisol or corticosteroid in mice, which many of you will know to be associated with stress. So this projection from the MPOA to the PVN, when June takes slices of the brain and does electrophysiology, puts electrodes in to record these neurons, when he stimulates them, he sees a suppression of their activity. He also then looked at the activity of the CRH neurons using in vivo fibre photometry that Rosie talked about, and showed that in mums that are lactating, when they're with their pups, there is less CRH neuron activity in the PVN. This led us to believe that perhaps this projection is helping to increase stress resilience in mothers. Now, Alice, an honours student of ours last year, started out to test this by looking at stress resilience in mums compared to virgin mice. She used a white box. This is the home cage, and it has a little speaker in it. And it plays a white noise, which is essentially like static when a TV has no signal. It's just an irritating fuzz, and it doesn't hurt them. They just don't like it particularly. So she compared virgin mice with lactating mums that show greater stress resilience. And you can see before the white noise, the corticosteroid is low, with a nice increase after the white noise. But this is severely blunted in lactating mums, showing greater stress resilience. And we believe that projection from the MPOA to the PVN is involved. Is it reducing anxiety also? Now, this is a test where the mum is placed in a box that she is well acclimatised to, and a door is lifted where she discovers her pups out in an open arena, which is a little bit worrying for her, a little bit tentative about going out. An anxiogenic environment, you might want to call it. And she found that in lactating mums, they are far less worried about it and will go out and interact with their pups far more readily, suggesting to us that in mums, when there is activation of the MPOA to PVN projection, they show less anxiety. So now we wanted to know what changes are happening in the brains of mums suffering from postnatal anxiety. Why is this usual circuit not functioning as we would expect it to? So our overarching hypothesis for the project was that in mothers, activation of the prolactin-sensitive MPOA to PVN neural circuit conveys stress resilience, and that disruption of this pathway leads to postnatal anxiety. So to do that, Rosie touched on the model already. We use a limited bedding and nesting model of postnatal anxiety. Just a reduction in their nesting material and the bedding that they're on leads to slightly disruptive behaviour, and we'll show you some of the results. Behavioural testing that we use, generally a pup assay in the home cage. It's just her home cage. They put it in a box and the pups are reintroduced like you saw in Rosie's videos. The barrier climb, she also showed you, of course, the white noise court test. And an open field where if the mouse is feeling more anxious, they will tend to stick to the safer perimeter. If they're feeling less anxious, they'll explore all around in the middle. And also a T-maze, which is slightly anxiety-inducing, just a harder task for the mum to go and find her pups. And then the last one, which I quite enjoy, and I think the mice do also, is an elevated plus maze. They're elevated off the ground, and there is an open arm here with no sides and a closed arm. If the mouse is more anxious, it will spend more time in the closed arms and less time in the open arms. They're placed on the maze and let run. A super adventurous mouse will run all over the place and sometimes dangle off the arms. It's kind of a playground for them. Anyway, that gives you an idea. So we've been testing the model out a little bit using these assays. And in a home cage assay, we see fragmented maternal behaviour. They will go to and away from pups frequently, disrupting their breastfeeding for the pups. And they will also show some less than positive behaviours, such as kicking. So in the LBN model, we see a greater level of pup kicking. The pups are not harmed, but it's not a particularly positive behaviour, and it suggests to us that they are more anxious. If you look at the pup weights during the first 11 days of lactation, the pups are receiving fragmented maternal care, less breastfeeding, and as a result they gain less weight. they're still surviving fine, they're just not receiving as good a care as they would normally in a mouse that is experiencing anxiety. So we have three main objectives. The first is to characterise what the anxiety-induced changes are in the PVR and CRH neurons. And we are doing this using the in vivo fibre photometry, where we can use sophisticated mouse lines and genetic tools to introduce a fluorescent indicator of neuron activity. And then we're looking at does postnatal anxiety change the activity of these neurons. So we're using the limited nesting and bedding model. 2 days after the birth of pups, they're introduced onto this paradigm, and then we can test their maternal behaviour and their anxiety, and also their response to a white stress noise. Then we want to look at what's driving the changes that we expect to see in the PVR and CRH neurons. And we're anticipating that the MPOA neurons, specifically projecting to the PVN, will show reduced activity. Remember, Rosie's seen that when pups are interacting with, when the mum is interacting with the pups, there is a huge activation of the MPOA neuron activity. We expect to see in a limited nesting and bedding model of postnatal anxiety that the activity of those neurons will be reduced. Then objective 2 is to further tease out the role of this projection from the MPOA to PVN, and to confirm its activity is involved in imparting stress resilience by shutting it down. We can do that using optogenetics, Rosie has already talked about, shining a light onto specific molecules that are introduced into the prolactin sensitive neurons that project to the PVN. We can switch them off. In a normal mum, without reduced bedding and nesting, she should be quite happy. But if we turn off those neurons, we hypothesize that we will see anxiety introduced, confirming that this neural population projecting to the PVN is very important. And then our final objective, really the icing on the cake, is to use a drug-targeted approach to activate the neurons in the MPOA that project to the PVN, to rescue mums that have anxiety through limited nesting and bedding from anxiety-like behaviour. So they will be introduced to this model when their pups are 2 days old, and then given continuous chemogenetic activation of their MPOA to PVN neuron population to hopefully rescue them from anxiety and to pinpoint that we are looking at a potential target for therapeutic strategies. That's mums, and all of this we're working on at the moment, so I don't have any data other than what the preliminary model data we've shown you. Last two or so years I've been working on dads. Anyone that's had a positive father role model in their life will know how important that is and how rewarding that is for the father. But 10 to 25% of dads in New Zealand also experience anxiety or depression, and yet there is limited funding for this, and it's received limited attention from both researchers and clinicians. We have a mouse model that is ideal, as mice also find pups hugely rewarding to interact with. So I'm going to show you a video of a mouse with fibre photometry, and here's a fibre optic that is in his nucleus accumbens, which is an area we know is involved in reward, and it will show you the dopamine release when he interacts with the pups. I think it's also on 2 times, quite fast. You can see the dopamine going up in response to those pups. But we don't yet know the neural circuits driving this in fathers. We would predict that it's the MPOA also, and we know there are prolactin-sensitive neurons in the MPOA as fathers also. Despite them not lactating or having young, they have prolactin, and what's it doing? It's supporting their parental behaviour. So this is a video of a mouse, a father with a fibre optic in his medial paroptic area. And it will show you the activity of those neurons every time he interacts with his pups. It goes up, and every time he goes away or stops interacting, it goes down. Possibly better than a mum. And this will go on for many minutes. And eventually, if I run this video long enough, he'll pick up the pups and take them back to the nest just like a mum. Dads don't show this behaviour, virgins don't show this behaviour at all. And if we go on, I've accumulated a lot of recordings from mice that do and don't show caregiving behaviour. And just watching the mice, as you did then, I can predict whether they're going to be caregiving dads or not be interested in the pups, purely on the level of activity that we see in their medial paroptic prolactin-sensitive neurons. So these are the caregiving mice here with a nice sustained long increase in MPOA activity just upon the first pup sniff. And those that aren't interested and don't show caregiving behaviour, it's quite truncated. And we've modelled this, and we can predict with 90% certainty that they will or won't show caregiving behaviour. And remember, these neurons, some of them project to the PBN. So the key question now is, can stress impair parental behaviour in males as it does in females? And we found that we have, purely by accident, found that we have a model where if we group house males, which is common, but if we group house males together, it's a model of social stress, and they will show far less parental-like behaviour than males that are housed individually. And so our prediction is that stress is disrupting the signalling of the MPOA to PBN neurons, as we think is also occurring in females. And we've been investigating that, and I have Vivian up the back, so fourth year student of ours, with some promising results already. So she's been using optogenetics, not to silence, but to activate the neurons which project from the PBN, from MPOA, to the PBN. And she's already found that in mice that are stimulated, they show less time displaying digging and anxiety-like behaviour. It's very promising to suggest to us that, as we suspect in the females, males also have a functional projection from the MPOA to PBN, which can be targeted to alleviate anxiety in dads. So while we're not experts, we're basic scientists, and we can't really help you directly with your questions about anxiety in mums if you're suffering. There are a number of really good resources which I've pulled out for you that I think the Neurological Foundation will share. But first and foremost, get in touch with your GP or your midwife. It's quite normal, 1 in 5 women who are suffering. There are some great resources that I've looked through from Plunkett, Healthify, and there's some really great videos with personal stories on there. Warning, you may cry, and they're quite confronting. And all of these have good rundowns on the symptoms that you may be experiencing, the kind of help, where you can go, etc. So with that, thank you. Ngā mihi nui to Neurological Foundation.